Hello everyone. All over the world, pathogenic bacteria disrupt life and cause multitudes of diseases through their use of specialized proteins. Hello, my name is Charles Wise, and along with Dr. Amanda Storm, we are investigating an unknown protein found in Staphylococcus aureus. Staphylococcus aureus is a pathogenic bacteria that infects millions each year and is normally known for its ability to develop drug resistance. Our research builds off of investigating unknown proteins that have had their structures modeled to understand their function. To identify a possible protein function, there were 3 assessments that required online prediction programs. These assessments were 1. Sequence analysis, 2. Sequence alignments, and 3. Structural features. Based on the sequence and structure, the programs were able to predict what this protein most likely resembled. The top predictions were that it attaches to the cell within the periplasmic space, and it being a unit of an ABC transporter, specifically the unit that binds the substrate and moves it into the transmembrane transporter. The substrate was also predicted to be nitrate. So in this image right here, this is an ABC transporter. This is a substrate binding protein. And what happens is substrate would be here, it binds to this protein, the protein then lines it up with the transmembrane transporter, and then that is then taken into the cell. The rest of these images build confidence in this hypothesis. The first one is the operon that this is a part of, and operons are sections of genes within a bacteria that are all produced into protein at the same time. All sections or all proteins of an ABC transporter can be seen in this operon, which builds confidence that this is likely a part of an ABC transporter. The second image up here is a multi-sequence alignment with our protein against known nitrate binding proteins. These highlighted regions are annotated regions that are known to bind nitrate. For our protein, there are 3 conserved or like residues that are the same in the nitrate binding proteins and only 2 differing. This means that this is very similar to nitrate binding proteins. This third image here is a superimposition of our protein against a known substrate binding protein, another ABC transporter system. Our protein is in purple and the opposing is in yellow. This is showing that they are very likely and similar. Doesn't mean that they are the same in function, but this is likely the substrate binding portion of that ABC transporter and not something else like the ATPase or the transmembrane transporter. So in conclusion, we think that this is the substrate binding part of an ABC transporter that is helping transport nitrate into the cell. We hope to use programs to actually model this interaction and do an experimental wet lab. And then we hope to finish the project by publishing a paper about all the findings. Thank you very much.